Hello all. Please let me know when I'm ready to begin.
Just wait, sir. Just wait, sir, please.
All right.
Good evening, everyone, and a warm welcome to this webinar, an international webinar jointly organized by the Departments of Botany, Physics and Zoology in collaboration with IQAC, Fatima Mada National College, Kollam. Now, let us invoke the presence of God. Molecular tracers paving the way for the new age of Theranostics and a personalized medicine. With the advent of new molecular imaging and therapeutic agents, a world of possibilities have opened up for the targeted diagnosis and treatment of many diseases, leading to personalized medicine. This talk looks at the field of nuclear medicine and molecular imaging sciences in the light of newer radiopharmaceuticals, detector technologies, clinical research, and applications that is changing many patient lives for the better. On this auspicious occasion, on behalf of the management, principal, IQAC, and all Fatima family, a cordial welcome to our eminent speaker, Dr. Remo George, Assistant Professor, School of Health Profession, University of Alabama, USA, for gracing this event with his presence. And a warm welcome to all for joining us for today's webinar. Now I call upon Dr. Cynthia Michael, our Vice Principal and Head of the Department of English. She is a person highly skilled with excellent academic expertise, vibrant and dedicated for the overall development of the college. I kindly invite Dr. Cynthia Michael for the inaugural address.
Good evening, one and all, and good morning, Dr. Remo George. Congratulations to the PG and research departments of Botany, Physics, and Zoology. I think it's the first time that we are having a program organized by three departments together. Normally, we have just one department organizing a program. And congratulations to the team IQAC of Dr. Shaiju, Dr. Sijin, and Bridget Thomas, and Dr. Nisha, and all the team members of the IQAC for organizing, what you say, not one program, but nearly every day we have one or two organized by the IQAC now. Now, this webinar, I understand, has great relevance in the medical field. From a layman's perspective, Theranostics is a combination of the two terms, therapeutics and diagnostics. And it's also used to describe the combination of using one radioactive drug to diagnose and the second radioactive drug to deliver therapy to treat the tumor. So in this treatment and in the cure of cancer, this is very relevant. Now we have got a very eminent resource person, Dr. Remo George, assistant professor from the University of Alabama, who will enlighten us regarding this. So I welcome Dr. Remo George to our institution also. Thank you, Doctor, for joining us today. On behalf of the principal, Dr. Jojo, I formally inaugurate this webinar, Molecular Tracers, Paving the Way for a New Age of Theranostics and Personalized Medicine. Thank you. Thank you, one and all.
Thank you, Dr. Cynthia, for your inspiring and enthusiastic words. I feel proud to invite Reverend Father Abhilash Grigori, our pro manager. He's a person who is highly principled, practical with definite goals, and a constant motivator. Father, I kindly invite you to deliver the felicitation.
Sister Father will join soon. We can bypass that.
Okay. It's time to know the chief guest of the day. Now I call upon Dr. Sinilan, Assistant Professor, Department of Botany, to introduce our speaker. Dr. Sinilan, please.
Good evening to all. I think it's a privilege for me to introduce my friend Dr. Remo George, and we are very glad to get him as the person who delivers the talk today. So Dr. Remo is now working as an assistant professor in UAB, that is University of Alabama, in the Department of Medicine Molecular Imaging Program. And then he has done some clinical practice over 10 years in Indiana, then Michigan, prior to his joining the School of Health Professions as a faculty member. So he has extensive experience in nuclear medicine and its procedures, then radiopharmaceuticals, and then PET applications. And he's also a US nuclear regulatory commission licensed medical therapy safety officer. So Dr. Ramo has received his master's. He has awarded bachelor's in zoology, and after that he got his master's in biophysics from Mahatma Gandhi University. And actually, I met this person there in Mahatma Gandhi University. And after that he went for a program, a diploma in this Baba Atomic Research Center. And then he completed, after joining University of Alabama, he did his PhD there. And then he completed his PhD in biochemistry and molecular genetics from the University of Alabama at Birmingham in 2014. And his current research interests involve the use of nanoparticles for modulation of free radicals in radiation therapy and the upregulation of sodium iodide symporter for theranostic applications in hepatocellular and breast cancers. I hope this will be an interesting talk, and I wish all the best to all the participants and especially Dr. Remo. Thank you all.
Thank you, Dr. Sinilal. It's a matter of pleasure and privilege to invite today's resource person, Dr. Remo George, to deliver the keynote address. Dr. Remo George, please.
Hello all. Thank you, Dr. Mary, Dr. Lal, Dr. Cynthia Michael, Dr. Shaiju, for having me here. And I also want to thank everybody for taking the time to be at this late hour, to be at this presentation. Let me try to share my screen here. I hope you are able to see the screen.
Yes,
yes, sir.
Okay.
Yes.
So my talk today is on molecular tracers and how it helps in paving the way for a newer age of Theranostics, and which leads into personalized medicine. Here are my learning objectives. I'll try to define molecular imaging in a very simple term. I'll try to describe how some of the SPECT and PET procedures fit into molecular imaging. I'll try to give some examples of molecular imaging procedures in clinical practice or under development, and I'll try to introduce how molecular imaging feeds into personalized medicine and patient management. So let me kind of go over what is molecular imaging. It is a way of visualizing, characterizing, and measuring of biological processes at the molecular and cellular level in humans, as well as in other living systems. So it's the visualization. It's a way of seeing the processes. Not only that, visualizing the processes at the molecular level as it happens within the biological system. Apart from that, it also involves the characterization, kind of visualizing the distribution pattern, visualizing the rate of change of these molecular signatures as distributed throughout the biological system. And a third paradigm is to help in the measurement of these changes that's happening at the molecular level. So we have been traditionally using this at the cellular level, but here we are doing this at the system and organ levels and the organism level. So as we just mentioned, it uses various strategies to image, which is to visualize the body's biological processes. So we know that changes in the structure of the body happens quite later down the road, which usually is quite a bit later after the changes that has happened in the physiology and the biochemistry of the body. So here we are trying to capture the changes quite early on as it happens within the biochemical system. So one of the important molecules in the body is glucose, which is used to obtain energy for the body's functions. So some of the biomarkers that is used to capture these changes in biochemistry is intimately involved with major functions in the body, like let's say, for example, glucose metabolism or nucleotide metabolism and so forth. So some biomarkers like FDG or fluorodeoxyglucose or fluorothymidine and so forth helps visualize these changes at the molecular levels. Usually, a patient gets to know that they have some kind of tumor or cancer when they feel a lump, or when they palpate an area and they feel as if something is not right. That's when they go for tests and so forth. Now, they could do a CT or MRI or ultrasound, and that kind of gives an idea as to the structural aspects of the disease. Now, utilizing such biomarkers will help in targeting the disease at the molecular level. So there are multiple modalities that look at diseases at the molecular levels, and this would include MR spectroscopy, bioluminescence imaging for gene expression. Ultrasound has some targeted contrast agents that does that. Molecular imaging, the strength of molecular imaging is in its ability to characterize as well as measure these processes. So it helps to assess the type of biological activity that's associated with the disease. It helps to compare normal cell growth and to distinguish it from the abnormal growth. It helps to estimate rates of cellular changes and to quantify those changes. So as we mentioned, these imaging strategies helps to measure and quantify the biological activity. And this involves extraction of data from these images and using software to quantify the change using parameters like standard uptake values to monitor the change over a period of time. So the benefit of this is especially evident in treating cancers, where you could characterize and measure and quantify the disease prior to treatment, and then administer therapy, and then measure the change over time to see if the therapy is working or not. If it's not, then a change in therapeutic regimen is called for. So nuclear medicine is basically the application of radioactive nuclides or radioactive tracers to image physiological processes in the body. Long time ago, way back when it started out in the 1950s, it's one of the byproduct of the Manhattan Project. We know that it's following the World War II, it ended up in the dropping of the atom bombs that ended the World War. But one of the byproduct of the Manhattan Project was the peaceful use of radionuclides in medicine and in research. So as a byproduct of that, radioactivity was used to diagnose and treat thyroid diseases. Radioactive iodine was administered to patients, and iodine has a propensity to get localized in the thyroid based on its activity And if certain parts of the thyroid does not localize the tracer, it means that it could be a cold area and which could be prone to disease. So it was a quantum leap in looking at physiology. And so images like these of the thyroid helped physicians to diagnose thyroid problems. This also led to treatment using iodine, different types of radioactive iodine. Some would emit gamma rays, which was specifically used to diagnose the disease, while some radioactive iodine, like iodine-131, would emit beta particles, which is used to destroy thyroid cells or destroy cells. And that could be specifically given to destroy cancer cells of the thyroid, which is very effective to treat cancers. So that was a long time ago. Images like these were obtained by rectilinear scanners that physically stayed on the patient neck, and that would literally scan in a rectilinear manner, dot by dot, the counts that came out of the thyroid. And then later on, it progressed to further radio tracers. One of the workhorses of, the workhorse of nuclear medicine is a radiopharmaceutical. Since I believe the majority of you have an idea as to what a radionuclide and a radio tracer is, I will condense my introduction a little bit. Allow me to share a screen for my whiteboard here. So a radiopharmaceutical has two parts: a radionuclide part and a pharmaceutical part. The radionuclide is the radioactive atom, and it is tied to the pharmaceutical part. So this pharmaceutical part can change. And if, let's say, the pharmaceutical is MDP, or methylene diphosphonate, and it's tied to a radionuclide called technetium-99 metastable, and if we inject this into the bloodstream, the pharmaceutical part, the MDP, would go into the bones. And wherever MDP goes, technetium would go with it, and it would light up the bones based on how well the bone is working. And that would be the basis of looking at the bone physiology. And that kind of imaging was done in the 70s and 80s. It was mostly two-dimensional whole body imaging with a gamma camera. And then later on, newer tracers came into the market, which were looking at specifically the heart, the kidneys, the liver, and the brain. Later on, after 2D imaging gave way to three-dimensional imaging, where a gamma camera would go around the patient and obtain multiple images of the organ. And this gave an idea as to the three-dimensional working of the body. This later paved the way for further development of the imaging area, including the development of 3D modalities, 3D imaging fusion modalities. So there was a tremendous leap in the amount of radio tracers that was available. And this brought about fusion of multiple modalities, including SPECT, which is imaging of the physiological radio tracers, SPECT with PET, SPECT with CT imaging. CT is computed tomography, PET, positron emission tomography, with MRI imaging. So multiple modalities start to fuse together to give both functional information and structural information. Traditionally, nuclear medicine have been looking at the function of the organs, including function of the bones, how well the bones worked, how well the heart worked, how well the heart pumped, how did the kidney filter the urine, how did the gallbladder empty out the bile, and things like that at the organ system levels. Later on, in the late 1990s, in the 2000s, and more so in the latter part of the 2000s, molecular imaging took precedence and has been leading the way now to look at diseases not only at the organ system level, but at the cellular level in vivo. So as we mentioned in the past, anatomic imaging was the norm with plain film, CT, MR, and ultrasound. And then came more functional imaging of the body using angiography, Doppler, nuclear medicine, PET, MRI, and so forth. And now we have hybrid imaging of PET-CT, SPECT-CT, PET-MR, and it is all leading into molecular imaging using nuclear medicine, PET, SPECT, MR spectroscopy, optical imaging, PET-MR, and contrast and enhanced MRI, ultrasound, and CT. PET-MR is the fusion of positron emission tomography and magnetic resonance imaging. We at UAB have a PET-MR system, which is actually one of the only systems here in Alabama, but this is catching on pretty fast. Now, the future molecular imaging modalities would include optical imaging. The ease of labeling with fluorescence and the absence of radiation issues is a strength of fluorescence imaging and bioluminescence imaging, but it's much more apt for animal research applications because of its low strength or low keV of its photons. There's targeted contrast, enhanced ultrasound, bioactive MR contrast agents, and PET-MR is very good for the molecular imaging of brain neurological processes. So traditionally, as we had mentioned, nuclear medicine imaging focuses primarily on the images. The physicians interpret these images via the patterns and the appearances of the tracer in the patient. Physiological processes had the greatest, traditional nuclear medicine has physiological processes as an emphasis at the organ system level. And the clinical process, the clinical research traditionally in nuclear medicine has been in humans, which has its issues. However, when we go into molecular imaging, a lot of possibilities have started to evolve. Now we can extract data from the image, the tracer data from these images, and it can be analyzed and can be interpreted via quantitative mechanisms. So quantitative mechanisms are reproducible. It is less subjective to the viewer or the interpreter, and it gives reproducible data. So, and also to quantify disease processes has its benefits. It can be correlated with the physiological processes as well. And it also opens up preclinical research in the realm of cells and animals. So, which brings to the concept of bench to bedside translational research. So now more and more labs are involved with these kind of translational research, which sets the stage for clinical trials later in humans. So what are molecular imaging agents? These agents are probes that lock in on biomarkers of disease processes that helps to visualize, characterize, and measure these processes in living systems. One of the classic the classic example for molecular imaging agent is the FDG, F18 fluorodeoxyglucose. This is a glucose analog. As you can see, in the second carbon atom, the hydroxyl group is replaced with a fluorine 18 group. So basically, I'm not going to go into the biochemistry of this. The FDG, as it can cross the cell membrane. However, once it gets into the cells, it gets locked in as it's converted from FDG6 phosphate. From FDG to FDG6 phosphate. And in cancer cells, due to Warburg effect, the cells are constantly thirsting for glucose, and it takes up more of this FDG, and the cells cannot distinguish between regular glucose versus FDG, and it takes it up and it gets locked in, and those areas are specifically lighted up, if you will, and those areas can be quantified. Similarly, with regard to FLT, this is specifically used to track DNA metabolism, and in cancer cells, these thymidine, fluorothymidine analogs can be used to measure the turnover of these labeled thymidine analogs. So how would the images look like with these molecular tracers? They look totally different with different types of tracers. As you can see, this is the fluorodeoxyglucose. Some of the areas, this would be a normal person. In a normal person, the areas that light up would be the brain, the heart, and some of this is excreted through the kidneys via into the bladder. So these areas are the largest consumers of glucose. As you can see, brain takes up a lot of glucose, similarly the heart. However, if you look at the distribution of FLT, you can see that normally a lot of tracer is taken up in the bone marrow. So a lot of this is excreted through the kidneys into the bladder. So just want to touch upon what is a biomarker. It is a characteristic which is measured and evaluated as an indicator of normal biological processes. It can also be used to trace pathogenic processes, and basically it can be used to as a pharmacological response to therapeutic intervention. So some of the biomarker targets that is under development and that can be used is the various pathways that happens in the body, including various receptors that are involved in signaling pathways, intracellular as well as extracellular targets. Oxygen can be tracked. The rate of utilization of oxygen can be tracked. The pH changes in the body can be tracked. New growth can be tracked. So some of the biological processes measured with molecular imaging include currently the glucose usage, the proliferation, the amount of proliferation of cells, amount of apoptosis that's happening, especially in cardiac diseases, in hypoxia, angiogenesis, new growth after revascularization therapies, and so forth. Some of the biomarkers that are under development include biomarkers to specifically target infectious processes. Some can target T cells, especially killer T cells, MR and muons for metastatic imaging. Certain intestinal adenomas can be lit up specifically by activatable fluorescence using certain antibodies. So all of those come under, are being looked at in the paradigm of molecular imaging. So how are molecular imaging agents relevant to clinical medicine? It helps to optimize patient care, and it improves the probability of a successful outcome for the patient's treatment. So if you look at oncology, it helps with early diagnosis. It helps to visualize the location, not only the location of the disease, but how much the disease has spread throughout the body or the extent of the disease. It helps to characterize, to determine the choice of therapy. And I would argue that this is one of the most important aspects of molecular imaging, is that it helps to determine choice of the therapy tailored to the patient's genetic profile, physiological profile, and in general, the patient as a whole. It helps to monitor and evaluate therapy, and it helps in guiding drug development. It has its applications in cardiovascular diseases. So the molecular imaging biomarkers can target ischemic injury areas, apoptotic, to look for apoptosis, looking for heart failure and left ventricular remodeling, thrombotic diseases, atherosclerosis, development of vulnerable plaques in patients, agents that specifically target these regions and light up these regions for intervention. Angiogenesis following revascularization therapies, cardiac arrhythmias, and as well as transplant rejection. In neurology, MI is playing a significant role in evaluating tumors. We have now agents that specifically lock on to the amyloid plaques in Alzheimer's that can specifically target the tau proteins. We have agents that help diagnose Parkinson's disease, seizures, various psychiatric disorders can be looked at as a function of brain function. So one of the critical areas where molecular imaging helps is in the treatment of various disease processes. So to give an idea as to how molecular imaging helps with disease processes and its treatment, is to look at how this can be applied specifically in certain cases. So shown here is a patient with a soft tissue sarcoma on the left thigh region. It's seen on CT as this area here. However, in a positron emission tomography using FDG, the glucose imaging, you can see that the extent of disease is much more than appreciated on CT. This patient is usually destined for surgery, but before that, they are given a short course of chemotherapy treatment, and then they are checked for its response. And that was done here. And you can see that the CT is showing, I don't know if you can see this, but the CT is still showing a lot of this disease still persistent after the short course of therapy. And the therapy, but then you can see that the PET CT So here in the left, the PET-CT was showing a lot of disease. The SUV, or the standard uptake value, was calculated to be around 10, which is a very high number to show that this is a very aggressive disease. Now, after that short course of treatment, the CT still showed a lot of area here. However, you can see that the area where of that disease has actually dropped drastically following that short course of treatment. In fact, the SUV dropped down to 1, which means that for this patient, that treatment is currently working. So, and it's working very effective for that patient. Even though, if you look at the structural modality for that disease, it did not show much of a change. However, with molecular imaging, that decision by that physician could be very sure-footed for them to continue with that treatment process. So this patient was had undergone surgery to remove these areas, and the treatment had continued with the chemotherapy regimen. So this kind of applications has brought into forefront a new paradigm, which include a PET-CT for initial diagnosis, followed by a brief standard therapy, and then another PET-CT. And now we have words like responder and non-responder. So responders can continue with the standard therapy after their interventions, and non-responders can be looked at with alternate therapies. That can save time, that can save lives. So molecular imaging provides a mechanism for translational research. So it helps with clinical trials for newer drug development using various biomarkers. It facilitates drug discovery and development, and it leads to the concept of personalized medicine. So a long time ago, in medicine, the impetus, long time ago, the impetus was to do no harm, because physicians, long time ago, they would say that if you intervene, then there's going to be side effects. So let's make things go as is. But later on, people realized that not doing anything is not going to work. So they started throwing everything at the disease, and that was the therapeutic era. So throwing everything at the disease to get it under control. But then it had tremendous side effects. But then sometimes that even ended up the patient's life. And now we have the age of much more of preventive medicine, where lifestyle changes and diet modifications and things like that are advised to patients to prevent, and the impetus is on preventing diseases. And also predictive medicine, you can kind of predict as to how the disease is going to progress, or if a person is going to get disease or not by analyzing genetic profiles and so forth. But the future is going to be personalized medicine, where individual persons, where the knowledge that two different people respond to the same medication in different manner. What is good for me may not be good for you. Two people respond differently to the same medicine, and basically the therapeutic intervention should be tailored to each person's unique genetic profile and physiological profile. So what is personalized medicine? Is a prevention, detection, and treatment of disease, taking into account a person's unique genetic profile. So it involves finding the right biomarker in targeting for the right target in the right for the right disease in the right patient. So that is targeted imaging, using targeted imaging to find the targeted therapy of the right drug against the right target for the right disease in the right patient. So which brings to the word Theranostics, which is using diagnostic modality that is to find out the correct therapeutic agent to arrive at the correct theranostic application to tailor a treatment based on the test results. So the poster child for Theranostics is using FES or fluoroestradiol and FDG to tailor a treatment. So fluoroestradiol is a F18. Fluoroestradiol is a tracer that binds to estrogen receptors. So HER2 or estrogen receptor positive cancer cells is being targeted here. So here you can see that this patient underwent FES imaging, and it's showing a positive area in the sternal region, which shows that this area is estrogen positive. That the cancer here is estrogen positive, which is on the spine. FDG, which is another molecular tracer that looks at glucose metabolism, is showing that this cancer area is highly metabolic. Now, so this patient then underwent anti-estrogen therapy, and following that, another imaging was done, and you could see that the FDG uptake is lower. So for this patient, that therapy will work. However, in this patient, you could see that FES is negative, while FDG, you could see that these areas are metabolically active. And after that anti-estrogen therapy, you can see that the cancer is still metabolically active. So for this patient, an alternate therapy is important. So, these kind of decision points are important for saving the patient's life. And Theranostics can play a great role in that. So you can see that this patient here responding to is FES positive, FDG positive, and is a responder after the therapy. While here you can see that FES negative, FDG positive, and FDG positive after therapy is a non-responder. So there has been a implosion of various molecular imaging agents being that is coming into the market, and there is ways to look at various molecular pathways and to develop disease probes. And I'm not going to go into details because I'm running out of time. So what does what does the future hold for our patients? There is improved diagnostic effectiveness. We can predict, monitor, and measure treatment responses. It helps to guide patient care, helps to determine most effective therapy to improve outcomes. It enables changes in individual patient care and management in real time. And also it saves a lot of cost in going around with unnecessary diagnostics and treatments. We in our program train technologists and technology scientists, helping to come up with newer modalities, newer tracers, as well as helping with diagnosing these and treating various diseases at the molecular level. So it helps to increase the number of biomarkers and contrast agents. It helps to increase the range of studies beyond the diagnosis. It helps to, it puts a greater emphasis on hybrid and multimodality imaging, and it helps in greater emphasis on quantification of disease processes. So I understand that we have audiences from various departments, including physics. I would like to go over some of the developments in the instrumentation aspects that helps with molecular, that helps with the progress of the molecular imaging. The workhorses of molecular imaging and nuclear medicine include gamma cameras and positron emission cameras. So to give an idea as to how these images are formed, here's the patient slice, and here's a camera. And just want to, some of you may be intimately aware of these things. I just want to probably maybe a review for you. So here's the gamma camera. It has a collimator. So here's the patient or an object with the tracer in it. There are gamma photons that are coming out, and only those perpendicular ones are allowed by these collimator. This is a lead collimator with physical holes in it that allows only perpendicular ones to come in. And these gamma photons hit a crystal detector, a sodium iodide thallium doped crystal detector, which produces scintillation photons in this detector. And these scintillation photons are then captured by something called photomultiplier tubes. These photomultiplier tubes actually convert these scintillation light photons into electricity. And these are amplified via something called diodes many times. And then they are fed into position and summing circuits that finally gives an XY location of the signal of these each and every count. So if a camera is sitting right at this angle in a patient, you can see that there's no counts, right? And then as we go over the organ, let's say this was a liver, the counts increase and then no counts. So this would be a count profile of a representation of the underlying tracer distribution. Now, as the camera looks at this tracer profile distribution all around the patient, then the principles of triangulation can be used to localize that activity within the patient in 3 dimensions. So that's kind of the basic in a nutshell how the computer determines the location of the underlying tracer. In case of positron emission tomography, there are, to understand positron emission tomography, the radionuclide emits a positron when it decays. The positron travels and meets an electron in the patient tissue, and they do a little waltz around each other, and they then fall into each other, and a annihilation reaction occurs, and 2 511 keV gamma photons or 511 keV annihilation photons are emitted. And these 2 511 annihilation photons travel 180-degree apart, and they're captured by detectors on opposite sides. So these multiple tiny detectors are arranged around the gantry. And if you look at each of these detectors, it is a, if you take out one of this detector, it's called a detector block, and it's coupled to these photomultiplier tubes that convert the photons into electricity, and they are then documented. So a lot of changes have been happening in recent times. One of this is the elimination of this PMT, the photomultiplier tube, which is coupled to the crystal detector that detects the photons. So this entire bulky photomultiplier tube, which has a photocathode where the photon is converted into electron, and then it's amplified. It is now replaced, or newer technology is now being being replaced with avalanche photodiodes and silicon photomultiplier tubes. This is called solid state digital photon counting. Basically, as photon hits these silicon photomultiplier tubes, it is directly converted into electrons with a gain. So, and also it can be configured into tiny strips, saving space, and also helps with localization, precise localization of counts. Another technology which is being developed is the light burst technology, which helps the development of PET-MR. The detectors, we have newer scintillation detectors, lutetium-based scintillation detectors, which helps with better scintillation so as the invisible gamma photon strikes these detectors, it would produce visible photons, scintillation photons. So how well a detector performs depends upon how fast these scintillation photons die down, so that it's ready for the next to receive the next gamma photon, right? So lutetium-based detectors are very fast scintillators and helps with this detection process and helping with better resolution. So as you can see here, let's say in bismuth germanate type of detectors, there's a larger spread in the signal, while in the newer type you have a much more better signal which can be concentrated and detected as to the precise location of the incoming signal. So 4 millimeter resolution for PET, positron emission, and SPECT is pretty good. 4 millimeter in the patient has a good timing resolution, better Compton recovery, so better scatter. Scatter is a big problem because it's the more the scatter of the incoming signal, poor is the resolution. So better scatter recovery. Silicon photomultiplier tubes help with better localization of the underlying signal. Thermal coupling, as well as we have the aluminum mounting plate, which is all arranged around the gantry now. Now, some other newer research into the detector technologies would include the mitigating the problem of parallax blur or parallax error correction. So as you can see, these are the detectors arranged in the gantry all around. And here is, let's say, a signal here. A atom decaying here. 2 511 keVs are going in opposite direction. But then it takes a little bit of depth for this detector to stop the 511 keV, correct? Because these are powerful photons, it can penetrate deeper. So that's why the detectors are much more longer than its breadth. So in the middle, it's okay. These detectors can stop the 511 keV, but as you move to the side, since these 511 keV is hitting the detectors in an angle, it will penetrate actually more and maybe stop by some detectors farther away. Now, the camera or the computer is going to think that, okay, the new line of response, this is the 2 511 keVs, it being detected is called a line of response. Now, the new line of response is here rather than here. So that introduces a spread of the full width half max, which means it's a poor resolution towards the edges. So newer research is based on how to mitigate these blur and towards the edges. So some of the newer developments in detector technology would include multi-layer detectors. So when you have multi-layer detectors, let's say some with different decay constants, which means maybe one layer has a slower decay of the scintillation photons, while the second layer would have faster decay. So that tells an idea as to the depth at to which these interactions occurred, and now you can tell the difference between something happening here versus something happening at the edge, and that tells as to where during modeling you can precisely tell within the system as to where the interaction occurred. So a various configuration of these arrangements is being looked at. So you can have with 2 decay constants, or you could arrange these 2 layers in a separate, in a slightly offset manner, so that when you do a flood with a uniform source, you could see that you could tell the difference as to where each layers have interacted with the incoming photons. Various other configurations would include multi-layer detectors. You have multiple layers of detectors so that you can precisely tell where the interaction occurred, at what depth interaction occurred. And this is made clear by certain light sharing methods and using different reflector arrangements so that these areas can be distinguished. Other depth of interaction measuring technologies would include having these stacked layers of silicon photomultiplier tubes between these detector layers so that individual layers are immediately distinguished. Detectors can be placed on either side so that if the interaction occurs in one side, there'll be signal will be more on that side versus signal being less on the other side, and so forth. A monolithic detector with underlying pixelated photomultiplier tubes, or position sensitive PMTs as they are called, the avalanche photodiode arrays, which are used to look at individual areas which can pinpoint individual areas where the detection occurred. And the centroid of those detection processes can be analyzed. And a lot of this involves mathematical modeling as well. To get much more precise details as to the depth, you could have crystals which are separated axially or which are cross-stacked to distinguish this depth. Another configuration is the DMICE detector. This is a offshoot of the from the research from the Large Hadron Collider in the CERN experiments in Switzerland. Where these two detector crystals are coupled, and in between there would be a triangular reflector, so that if the photons came in and interacted at the top, in that case the two detectors would see that interaction, and you have a larger spread in the output. While if the interaction occurring at a different level at the base, then the two individual detectors will have a different type of output response. Another kind of configuration is coating the top of the detector with a phosphor, so that if interaction occurs at the top, you have a larger pulse output, while at the bottom, since there's no phosphor coating, there would be a smaller output. Still, other configurations include, if in a array of detectors, if the interaction occurred at the base, less of these detectors would see the interactions, while if the interaction occurred at the top, more of these detectors, as the light is spreading due to reflectors, more of these detectors will be seeing this. And also you have these multiple kind of these triangular teeth strips of reflectors that would help with, again, detection of the depth. So there are these different types of research that's going on to better the systems, and such systems have helped with advancing molecular imaging tremendously and bringing in very MRI grade resolution to analyzing physiological and molecular processes in the cell, and which is big for diagnosis. So I also know that we have this is a talk with the plant biologists, botanists. So molecular imaging has been used in plant sciences as well, especially in the process of stress detection. Plant stress detection is a critical area for improvement of crop yield. Traditionally, we have been using metabolomics, but then it would involve destruction of the plant in biochemically analyzing the processes. Fluorescence imaging, thermography, visible near-infrared reflections, and so forth are non-destructive, but as far as resolution, they have lesser accuracy. PET and SPECT have been used to look at the physiological processes. And also it has helped with quantifying processes. So to give an example, a study that came out in 2019 by UC Davis, I know this person, Simon Cherry, he's one of the pioneers in molecular imaging. So they have shown how PET can be used as a promising and practical candidate to complement traditional analysis method for plants. And they have shown insights into the systems level salt transport mechanisms in intact plant. I recommend that if you're interested, you could read on this article. So in all, molecular imaging has been in the forefront of nuclear medicine, and in 2008, our society changed its name from Society of Nuclear Medicine to Society of Nuclear Medicine and Molecular Imaging to include these paradigm changes. So nuclear medicine is an integral part of molecular imaging, and molecular imaging makes personalized medicine possible. I apologize that I went a little over time. I was told it's a 1-hour talk. So that concludes my talk.
Thank you, Dr. Remu George. It was quite informative and beneficial for all of us. It was indeed an exhilarating experience which enlightened our knowledge on molecular imaging in radiopharmaceuticals and measure biological processes in living systems. Of course, the forthcoming revolutions of molecular theranostics is a broader, integrated view of personalized medicine. Thank you, Dr. Remo George.
Thank you.
Now the session is open for discussion and queries.
So there are 2 questions in the Q&A box. Can you please?
I guess there are 2 questions. I'll read the question.
Yes, I shall. The first one from Dr. Rose D. Lima. Interesting to know about the new age medical advancement. Altogether, it sounds very exciting and promising, but this personalized targeted medicine will not be available for a common man in near future. That's a question. Doubting the cost of this treatment will be exorbitant. Is this molecular studies for treating deadly diseases at molecular level will be time consuming? So these are the questions, sir.
Yeah, that's a good question. That is a matter-of-fact question. Molecular imaging has been very ubiquitous, actually, all around the world. If you look at, I think even at the community level, you have PET cameras now in India, in Kerala, I know there are PET cameras in Kottayam, in Trivandrum. We have been looking at FDG. We have been looking. Anybody with cancer nowadays has to undergo an FDG imaging to look at how the pattern of uptake of glucose in their bodies. So molecular imaging is very ubiquitous than what we may tend to think about. As far as molecular therapies, I agree that these are very pricey, very costly therapies, and sometimes beyond the reach of common man. But then that is the way with all therapies, I guess, initially. But later on, as technology progresses, it will definitely come down. I know for one of the common therapies that we do here at UAB is the neuroendocrine tumor. Is a lutetium DOTA therapy, which can cost close to like $40,000, which can be pretty exorbitant. However, it is important that the government helps with this process as well. If there is a way to subsidize these kind of therapies in some manner, that will definitely be a help.
So another question is again from Rosli Lelima. So what is the time period for diagnosis of very rare diseases for using this molecular tracing?
Diseases. So when you say time period, if the therapy or if the diagnostic agent is FDA cleared, which is in the US, it should be available right away. And some of these, so the radio tracer is injected. There is some amount of circulation time. It's usually in the realm of hours, and some of those rarely would take a couple of days. And then once the radio tracer reaches the target organ, it's usually in the hours. I can tell you about 80 to 90% of the tracers currently available. It's in the hours. And it can be characterized, and a diagnosis can be achieved in 1 to 2 days time.
So another question from Dr. Satish Kumar from Banaras Hindu University. It was, my question is, what would be the basis to decide the personalized medicine? Is it genomics? How long it may take to get it standardized in that case, as the genomic data can be so complex?
Yes. So when we say genomics, what I meant was we are not analyzing the genome. We are actually basing the treatment tailored to the patient's response. And what I meant by genomics was the patient's response is usually tailored to the underlying genomics of the patient. So we are not analyzing the genomics. We are analyzing the response based on the patient's personal genome. That's what I meant.
So no more questions in the question and answer session. Participants, if you have any questions, please record at the earliest. If you don't have, we will go to the next person. Sister, please.
Thank you, Dr. Remo George. Father Abhilash Gregory could not attend the webinar due to some personal inconvenience, and he has sent a video, and now let us watch the video.
Good evening to all. I am privileged to say a few words on the auspicious occasion of the international webinar on molecular tracers, paving the way for a new age of Theranostics and personalized medicine. I have to thank departments of botany, physics and zoology for conducting this seminar. See, I am a layperson and I don't even understand the heading itself. But I understand one thing, that how science comes to the day-to-day life of every human being. How science does not confine itself in the books, but how it makes our life happier, healthier, stronger, safer. So this is what the nutshell of this webinar, I think. And we are fortunate today to have the blessed presence of Dr. Remo George, Assistant Professor in the School of Health Professions, University of Alabama. Sir, I take this opportunity to thank you for spending your precious time for us so that we may be enriched through your words of wisdom. Once again, I thank all those who have worked hard to make this venture a fruitful one. Thank you, and may God bless you all.
Thank you, Father. Now one more question. I request Dr. Nisha to put forward the question.
Good evening all. Am I audible?
Yes.
Yes.
Okay. So at the outset, I wish to thank Dr. Remo. Yeah, can you hear me?
Yes.
Yeah. I wish to thank Dr. Remo for not disappointing either the three departments, as he was able to cover, aspects pertaining to every department. But one question that I would like to ask is, see, I was quite recently exposed to an incidence of tachycardia, and the remedy that they did for that was to subject the person to a radio frequency ablation procedure. Through your talk in between, I just came across a term where you mentioned an application regarding left ventricular remodeling. So could you please enlighten me regarding that particular aspect? How does nuclear medicine play a role or help in left ventricular remodeling?
Well, thank you for that question. That's a great question. We know that left ventricle of our heart is the largest chamber and the most powerful chamber. When we assess the function of the heart, it is the left ventricle's function which is the most important. Traditionally, we have been able to measure the function of the heart by its ejection fraction, how well the left ventricle contracts and pushes the blood out of the heart. Now, that ability of that left ventricle to pump depends upon how well the muscles of the left ventricle is working, or how well the muscles are perfused with blood. Now, in case of an issue with blood supply, let's say, for example, if that's the case, then that particular area of the heart would start to die off or would start to necrose because of lack of oxygen. And one way around that is to revascularize that area. And things like angiography or angioplasty. Angiography can be, it's a invasive method to look at blood supply to those regions. However, angioplasty is a way to revascularize that area, or to ablate that area, as well as to graft tissue into that into those areas. Some of the recent developments is to look at if we could use stem cell therapy to regenerate dead areas of the myocardium or non-working areas of the myocardium. So in that aspect, molecular tracers have been used to check for how well those areas are regenerating, which is something no other modalities can provide. How well the angiogenesis is happening in real time in those areas, which then can be used to, tracers can be used to check for how well the myocardium in general is functioning. So those kind of applications are being done in cardiology. I hope I was able to answer your question.
Yeah, hopefully. But am I audible still?
Yes.
Okay. So after this particular person underwent this procedure, what they said was that there wasn't any method, for a follow-up, because they couldn't have a follow-up. So it just depended on the luck of that particular person, and we just have to hope that the procedure was successful. But I understand that through this method, it is possible for us to find out whether the tissues have sort of regenerated. I hope I'm correct. So is such kind of a work going on in India? Is it do we have such kind of a monitoring in Kerala, to your knowledge? Are you aware of such kind of a work being done in India?
Yes. Now, with regard to that specific example which you presented, what I am understanding is that it is much more of a neuronal issue rather than a perfusion issue. So, with regard to those kind of treatments, I'm not quite aware of the use of molecular imaging specifically for the neuronal aspect of the cardiac issue. With regard to the perfusion aspect, I'm pretty sure, even though I do not have firsthand experience knowledge, but I'm pretty sure that larger research hospitals like All India Institute of Medical Sciences would have those capacities to look at issues like that. Again, I'm not intimately aware of if the specific things I mentioned is available in India. However, FDA, this is, as I mentioned, some of this is very new, and FDA has just approved agents to look at apoptosis in the heart. So it may not still be available ubiquitously, but going forward, we'll be seeing more of those.
Thank you. Thank you, Doctor. Thank you very much.
Thank you so much. Dear participants, feedback form is in the chat box. Please see it. It's time to wind up the session. Now I call upon Dr. Nirmala Jairani, Associate Professor, Head of the Department of Botany, to deliver the vote of thanks.
Ma'am, please unmute.
Can you hear?
Yes, ma'am. Okay.
Good evening, all of you. Molecular traces paving the way for a new age of Theranostics and personalized medicine. So we are happy. Really, I am happy to express my deep sense of gratitude to Dr. Ramo, Assistant Professor, for being with us to deliver his talk. Really, his talk was very inspiring and he has given a very clear picture about how the molecular imaging and therapeutic agents, a world of possibilities have opened up for the targeted diagnosis and treatment of many diseases, leading to personalized medicine. This talk looks at the field of nuclear medicine and molecular imaging sciences in the light of newer radiopharmaceuticals, detector technologies, clinical research, and applications that is changing many patient lives for the betterment. Really, we are happy to have you, sir, with us. I take this opportunity to express our deep sense of gratitude on behalf of the manager, principal, IQSC, Department of Botany, Department of Physics, and Department of Zoology. A great vote of thanks for sparing your research with us. Really, it was a very clear picture how your research has paved the way for the new world of therapeutic science, new world of science. So I also take this opportunity to say a word of thanks to Dr. Sinilal for arranging such a wonderful talk. And also, I express my sincere thanks to the IQSC team and also all the participants present here for being patient with us and also giving your wonderful support. Once again, I thank Dr. Ramo George for presenting a very wonderful topic, how the personalized medicine research has improved and being helpful for the patients. Thank you very much, sir. And thank you all the participants present here. Thank you once and all.
Thank you, Dr. Nirmala. So once again, I thank everyone, each and everyone who wholeheartedly participated to this webinar. And thank you one and all.
Thank you, sir.